So endocrine regulation of reproduction. In this section, we're going to talk about all the hormones from hypothalamus to the anterior pituitary and then to the other endocrine glands as well as our gonads, how the hormone regulates the reproduction. Okay. So here, this flow chart, you can see we have gone over this in the endocrine system. So in the early age, we have these hormones a lot, but at puberty is exactly what here happens. So during puberty, when reproduction starts, our hypothalamus release a releasing hormone called gonadotropin releasing hormone. Gonadotropin releasing hormone acts on anterior pituitary particularly, and that produce follicle stimulating hormone, luteinizing hormone. Both follicle stimulating hormone and luteinizing hormones have receptors on gonads. In female on ovary, and in the male it is on testis. And these gonadotropin, both follicle stimulating hormone and luteinizing, when binds with the receptor on the gonads, they produce hormones. So like in female, sex steroids will be estrogen, progesterone. In male, it will be testosterone. And then there is other cells, particularly in the testis and other follicle cell, they make another protein called inhibin. So these pulse-like release of hormone in the body gives negative feedback mechanism and maintain the concentration of these hormones always in normal concentration by negative feedback mechanism. So you can see here the sex steroid hormones, which is released from the gonad, whether it is the ovary or testis, they go and give negative feedback to the anterior pituitary and to the hypothalamus. Okay. Whereas there is another protein produced by gonad cells too, and that is called inhibin, and that can give the negative feedback to the anterior pituitary cells, particularly which secrete follicle stimulating hormone. So that's how this hormone is maintained like in a homeostasis in the body. And the main function of these hormones produced by the gonads is to produce gametes, particularly sperm and ova. Okay, that's how the hormone from the hypothalamus and anterior pituitary and gonads maintain the reproduction. Okay, onset of puberty. So puberty in male and female are slightly different age, like 10, earliest in female is 10, late in female, in the male is 16. So what brings the onset of puberty in us? That is the follicle and stimulating hormone, follicle stimulating and luteinizing hormone secretion from the anterior pituitary is high for the first 6 months of life, which is in the fetal life. And then what happens? If you remember the first lecture, then what happens? There is no, the hormones, particularly these hormones, they are very low till puberty. And during the puberty, the hypothalamus increase gonadotropin releasing secretion from the hypothalamus. That stimulates increased luteinizing hormone and follicle stimulating hormone, which stimulates sex steroid secretion from the gonads. And that drives change in male and female secondary sex characteristics, like the menarche, which is the first menstruation flow, then growth of pubic hair, axillary hair, and other male and female like body appearance is also under the influence of androgens, secondary androgens coming from adrenal cortex. So it is not only testosterone, estrogen, and progesterone that makes you male or female. It is other hormones which also adds of external body feature of male and female from the hormones coming from adrenal cortex. Adrenal cortex has three regions. You remember that. Zona glomerulosa, zona fasciculata, zona reticularis, which zona produce the hormones which help in growth of pubic and axillary hair.
Glandulosa?
Nope.
Reticulus.
Reticularis, the innermost layer. Okay. Okay, so growth and function of sex and age. So growth is spurt in male and female. You see here? In the x-axis, you have the age, and in y-axis, height gain in centimeter per year, so every year. When you see from the age 6, 7, 8, 9, 10, there is no growth, particularly in height and weight. And in female, you see, by the age of 10, all of a sudden, GnRH kicks in, and then they produce follicle stimulating hormone, luteinizing hormone, and then other characteristics, hormones. So female growth spurt, you see. Here it was 6 centimeter per year now, up to here, every year. Now you can see all of a sudden it is 9 centimeter per year growth. So this is called growth spurt. And then by the age of 12, 13, you see female start dropping the rate of growth. So by again 14, your growth is like only 2 centimeter per year. By the age of 18 or 17, pretty much none. So by the age of 18, you will not, whatever height you have that height. But male starts slightly later, like 13, you see, growth is spurt start, and it is maximum 8, 9. You see, at the age of 14, you have like 9 centimeter per year. And then for male too, after like 15, goes down. Okay, so that is the growth spurt in male and female. Slightly different. Female has early, faster growth than male. The means of reproduction in human and in other mammals are copulation. And this part is usually covered in the health education class and other psychology, psychology of the sexuality. But I'm going to touch base a little bit physiological change in the body during copulation or sex act of the sex. So there are 4 phases of human sexual response. And those 4 phases are excitation phase, plateau phase, orgasm, and resolution phase. So let me go first. Excitation phase is phase of excitation. Plateau phase is like you stay in that phase for a time being, and orgasm phase is the phase of ejaculation in both in male and female, and resolution is return of the resting phase. Okay, so what happens in each phase? Excitation phase is arousal phase. It's characterized by increased muscle tone, engorgement of sexual organs with blood, includes nipples, clitoris, penis, labia minora, and there is secretion, increased secretion in the vagina, that is lubricating the vagina. So excitation phase follows the parasympathetic stimulation. So when you have sensory input like visual, tactile, touching, hearing, visual, all kind of external stimulation activate parasympathetic nervous system. Then parasympathetic nervous system arousal or activation bring this all change in both male and female. Certain feature pertains to female, certain feature pertains to male. Once this is a phase in both male and female, then there is this phase remains in that case, plateau phase. plateau phase. Clitoris is partially hidden by swelling of labia, and areola continues swelling, and outer third of the vagina further engorges with blood supply and secretion. And then during the sexual intercourse, there is ejaculation in men, and in women, that is called the analogous contraction of uterus in the vagina. So you contract the floor of the pelvic floor, and contraction of the vaginal wall, and contraction of the uterus. That is the like analogous orgasm in the male and female. Orgasm is pretty much the this word explained like a you cannot describe it, you can feel it. That is orgasm is, okay? And then resolution is the everything. So this orgasm phase is the action of sympathetic nervous system. Doesn't it look like opposite? Usually what happens when you have sympathetic nervous system activation, that means everything is increased. But here, parasympathetic, do you know why? Parasympathetic increase blood flow in the genitalia. And you will see when we do the mechanism of erection in male, okay? So everything after the relaxation, after the activation of sympathetic nervous system, everything returns to normal, and that is called resolution phase. Body returns to pre-excitation condition, and then man enters in refractory period. Refractory period means after one sexual act, how long you have to wait to have ready for another sexual act. That is called refractory period. In male, there is a minimum 30 minutes, and based on whom you're talking about, it can be 30 years, okay? So that is the refractory period in the male, but it is not applied in the female. Female has not such a refractory period, okay? So that is the first part, the some leftover from the part A of the reproductive system lecture. Let's go to next. And then we will stop sharing this. Okay. So now the male reproductive system. There is a lot of things we have done, so I'm going to go over those things a little bit faster. In this section, this is the chapter 20, reproduction, particularly male reproductive system, which is our part B. I will go over organs of the male reproductive system. What are they? Control of gonadotropin secretion, how it is controlled. You know how it is controlled. We did the regulation of endocrine regulation of the male reproductive system or any reproductive system. Then we will talk about endocrine function of the testis, how testis act as the organ to make sperm, as well as also an organ which produces testosterone, which makes the male male, okay? Then we will talk about the processes of spermatogenesis, the steps in sperm formation inside the testis. Then we will talk about male accessory sex organs, which carry sperm from the testis up to the penis and then deposit in the vagina during the sexual intercourse. To do that, to transport the sperm, it is the penis organ which does that. And to do that job, the penis needs erection and then movement of semen through the penis. That is called emission, and then ejaculation, which is outside forceful expulsion of semen from the penis. That is ejaculation. And we will talk a little bit about male fertility. When you are married and you have a regular unprotected sex for a year and then you don't get baby, then you need to think you have some kind of primary infertility. And I will suggest first person to test is the male, not the female. Okay. So male reproductive system, this is the diagram. We have a mid-sagittal section of a male reproductive body. This is anterior part and this is posterior part. So the primary organ of the male reproductive system is testis, which is lying in the scrotum. This testis makes a sperm, and this testis makes also one hormone that is called testosterone. So hormone from here get into surrounding blood vessel and then circulate all over the body, whereas sperm from made from this testis pass into this comma-shaped tube that is called epididymis, and that epididymis then is continuous with the vas deferens or ductus deferens, which carry the sperm, and then this vas deferens or ductus deferens enters our pelvic cavity, and this hook around the ureter, you see this ureter, this side you can see this side is cut, so this goes around here like here, so it hook above the ureter, and then it comes back and slightly enlarge. That enlarged part of the ductus deferens is called ampulla of the ductus deferens. Then this ductus deferens becomes the ejaculatory duct. Once a gland on the side of the ejaculatory duct called seminal vesicles join the vas deferens, then it becomes ejaculatory duct. Then ejaculatory duct from both sides, you see this is the right side, left side, and right side is here. They come here and they join the urethra. Urethra is passing through the prostate gland in man, so this is called prostatic urethra. Then it runs through the urogenital diaphragm, we call them membranous urethra. And in during the membranous urethra, there is a small gland, you see, which is joining the urethra. What is that gland called?
The bulbourethral
Prostate.
gland.
Bulbourethral gland. You see, bulbourethral gland, this P-shaped gland is joining the membranous urethra. Prostate gland is here, joining the urethra here. We throw the small ducts, they don't join here. Okay. Now, once during the sexual intercourse, the sperm is here, stored in the ampulla. And when this ampulla contracts, the ductus deferens contract, because ductus deferens is a tube which has a muscle, so they contract. When they contract, they squeeze the seminal fluid and the semen. Seminal fluid and the sperm, they become semen. Then some prostatic fluid comes, some fluid comes, and they all add up. Then semen passes. But before semen passes, this P-size bulbourethral gland also produce some fluid which is rich in bicarbonate alkaline, and that clean the urethra, and then semen comes out. Okay, so the next organ is the penis. Penis has the two kinds of erectile tissue. The upper one is called corpora cavernosa, and the lower one is called corpora spongiosum, and urethra runs through the corpora spongiosum. Okay, so this is penis. You see there is glans penis and there is prepuce, which the skin covering it. So this is the primary organ, and all other organs are accessory organs of the male reproductive system. Okay, so which organ makes the sperm? Testis. So we need to study the histology of the testis. So here is the testis and the epididymis and the vas deferens. Vas deferens is covered by a lot of connective tissue, and the sac of connective tissue which is covering the vas deferens, a lot of blood vessel, nerve, lymphatic vessel. That cord which contains these all pipes is called a spermatic cord. Spermatic cord runs through the inguinal canal and get into your body. Okay. Now let's see the testis. So when we cut testis in half, what are we going to see? Inside the set testis, you see there is coiled small tubes. These tubes are called seminiferous tubules. Okay. These seminiferous tubules are distributed in the lobules. You see, each lobules have like 2 to 4 coil tubes. Okay, so this is the seminiferous tubules in the testis. Now, if we give a transverse section here, let's see if we cut here. Take one tube here and then we cut here. What are we going to see? We're going to see this one. Okay, so let me erase it. So when we cut, and then you magnify this one of the tube. So here is one complete tubule. There is another tubule. There is another tubule. Are you guys following me? Okay. So if we understand one of the tube, we can understand everything. So here is tube and there is space outside the tube, you see? So this is intratubular compartment, and there, this is small area. Let me point it. This is small area here is the extratubular compartment. Okay. Intratubular compartment is formed by the tube. So if you see the wall of these tubules, any tube take and then see the tubular wall, magnify it. So if it is a small tiny tube, and now I'm magnifying this tube, you see? And there will be like several tubes. We are focusing on one tube. The outermost layer of the tube is the basement membrane you see outside. And then second layer of cells are germinal epithelium here. The germinal epithelium are the spermatogonia. These spermatogonia chains divide by meiosis, first meiosis and second meiosis, and makes the sperm. When they make a sperm, you see their head are lying towards the basal area of the tube, and the tail is facing towards the adluminal. Adluminal means towards the lumen. Okay, so this is the lumen of seminiferous tubules. Are you following me? As you can see the spermatozoa. So these germinal epithelial cells makes the sperm, and to help these cells, in between these cells, there are other cells. We call them Sertoli cells. You're going to see that. They help germinal epithelial cell to make the sperm. Okay, we'll see that later. On the other hand, outside the cells here or here or here, you see some other cells. You see nuclei of 2 nuclei here. And those cells are called interstitial cells or Leydig cells. These Leydig cells produce hormone, and that hormone is testosterone. Okay, so here you go. Testis contains seminiferous tubules where spermatogenesis occurs, and interstitial tissue housing testis secreting Leydig cells. This cell produce testosterone, whereas some cell in between these cells here in the germinal cells, like if there is one cell, Sertoli cell, another cell, Sertoli cell, in the middle, cells will be germinal epithelial cells. Sertoli cells of ST cell, seminiferous tubules contains receptor for follicle stimulating hormone, which stimulates spermatogenesis, okay, which helps. These cells do not make sperm directly. They produce hormone, or other way, indirect way, they help germinal cells to produce sperm. Whereas Leydig cells has receptor for LH hormone, and LH hormone is coming from anterior pituitary, and that activates the Leydig cells to produce testosterone. But this testosterone circulate into nearby blood vessel, or they remain here. Blood brings through the blood, and through other, the protein produced by Sertoli cell will bring this testosterone here, because testosterone is essential for these germinal epithelial cells. Without testosterone hormone from here, these cells cannot transform into a sperm. Are you following? This is very important thing to understand. This hormone goes everywhere. It goes to your brain, to your muscle, to your body, to do other function. But when it comes here, this testosterone has a receptor on these cells too. They help these cells to transform into sperm. Okay. Okay, so this is the same thing we talked earlier. I'm not going to go here. Testosterone and age. Testosterone is highest during the first 6 months of intrauterine life. True or false? True. Testosterone is highest during puberty. True or false?
True.
True. Testosterone is highest at the age of 6. True or false?
False.
False. And in male, after age 50, testosterone start declining. The concentration of testosterone. Nobody knows exact reason, and it is not in everybody the same way. Some male will have drastic decline in testosterone after 50, but some people will have 70, 80 years and still not the same level of decline of testosterone. That's why a man can fertilize up to the age of 80 years of age. You have seen people 80 years age can fertilize. Okay. So there is no exact known cause, but not due to reduced GnRH, LH, or FSH, because these hormones level are high in that age. Okay. So I'm going to go in detail the spermatogenesis. So which cell produce sperm from the seminiferous tubule wall?
German.
Germinal cells.
Germinal cells. Okay. So in the wall, you have the spermatogonia. Okay. So germ cell that migrate from yolk sac during development become a spermatogonia or stem cells. So what happened during early age of fetal development? You see there is fetal sac, there is the yolk sac. From all the primitive cells for the future sperm or egg are in that yolk sac. And from there, they migrate to the developing gonad. In case of male, it goes to the primitive testis. Okay. And during that time, these gametes are spermatogonia. You see, spermatogonia are 2N. 2N means they are diploid cells. They have 23 pairs of chromosomes. Okay. They are diploid. And then they remain there. So spermatogonia replicates cells throughout life by mitosis in the wall of seminiferous tubules. So they are like continuing dividing. You see, from 2N, they divide. So what is the important? Why I'm saying arrow here and arrow here? They divide. Let me tell you how they work. So this is important to understand. Okay. So here, if you have one cell, they come here and they divide into two, A and B. And when they divide, what are they? This is 2N and this is 2N too. So I'm making here different way, A and B. This B remains here and A goes back in the wall of the seminiferous tubules. That's why you see 2N here. Okay. And significance of this one, you will see in future. So now let's see if you have a bank account where you have $100 and bank gives you a choice. Hey, every time you take out $100 from your account, you can make it 200 and you can keep 100 back in the bank and you can use that $100. Will you lose that $100 ever?
No.
No.
Okay.
So that's why every time you divide, you keep one inside the wall and one goes down, and that becomes enlarged inside with enlarged nuclear material, and that is called primary spermatocyte. Now, during the puberty, this primary spermatocyte cause the first meiotic division. And you know meiotic division is haploid. So they make two haploid cells, and that's why we call them secondary spermatocytes. This is after the first meiotic division. Then each secondary spermatocyte makes the copy of each and makes two from each copy, similar copy, and that is called second meiotic division. So second meiotic division is like just the replication of exactly what you have, and you make 4 spermatozoa from one spermatogonia. You keeping that spermatogonia, and then you are making 4. So you can see in the how many seminiferous tubules you have in the testis, and how many cells of a spermatogonia are in the wall of each seminiferous tubules in each section, and how many cells they are dividing this way, and how many they are keeping in the wall. It is like never ending number of sperm. They can make unlimited amount of sperm in whole male life. That's why there is a saying, a single male, healthy male can make as many as sperm that a single man can fertilize whole world egg, if each and every sperm is viable. Are you following me? But female, you will see when we talk about female reproductive system, it does not happen with the female. Female start with some 4 million, and by the time in your puberty life, female's puberty life pretty much stops at the age of 49, 50. So from like 16 or 14 till the age of 50, female in average ovulate only 400 eggs. Okay, that is this is called spermatogenesis. I'm going to talk about one more terminology here. So from spermatogonia to spermatids, this all process is called, this is the spermatogenesis. From here to here, from spermatogonia to spermatozoa is spermatogenesis. But the subset of spermatogenesis where you make spermatozoa from spermatids are spermiogenesis. You see spermiogenesis? So spermiogenesis is transformation of spermatids to spermatozoa. And this happens with the help of supporting cells, which is lying, like if it is one spermatogonia, there is another here and here. Each spermatogonia will be, let's see this, my mouse is spermatogonic cells, which forms a sperm. 2 cells on the side will be my 2 hands, will be my supporting cells, okay? So those supporting cells helps this process from, and you're gonna see that later, okay? So here you go. You see, this is the spermatogonia, 46 chromosome, 23 pairs, and there is one supporting cells. It's nuclei, And then this side, you can not see another cells. So you see all the stage of cells coming here. And then this, once spermatids are formed, then they slowly develop their head, tail, and then their excess cytoplasm. Excess cytoplasm is pinched off and taken by these supporting cells and digested by the lysosome enzymes inside the supporting cells. Okay. So spermatogenesis occurs in the wall of seminiferous tubules. Spermatogonia and primary spermatocytes are located in outer part of the seminiferous tubules. Spermatids and mature spermatozoa are located towards lumen. And these we talked about these things. I'm not going to read it. So you see, here is cytoplasm of the spermatids. Tail is coming out, and then you see whole cytoplasm is pinched inside coming, and then sperm is coming outside. Okay. Here is spermiogenesis. Is maturation of spermatids into a spermatozoa. See? So this pinch of cytoplasm is eaten by these supporting cells. Okay, so let's talk about the function of the Sertoli cells. So what does Sertoli cell do? Sertoli cell is supporting the Sertoli cell is supporting the spermatogonic cells to produce sperm. How they are doing? They are providing nutrition to the cells. They are helping in spermiogenesis. And spermatogonia are the spermatozoa are the haploid cells. And your immune system has never seen a spermatogonia or your egg cells because they are haploid. If T lymphocytes and B lymphocyte reach these cells anywhere and find them, they're going to attack them immediately and kill them. That's why to protect from immune cells, the Sertoli cells produce the blood-brain barrier. So what happens? Let's see, this is the one side of the Sertoli cells, and there will be another Sertoli cells. These wall of another Sertoli cells and wall of this Sertoli cells, they will have lateral cells attachment plasma membrane, and they create blood testis barrier. So there is blood testis barrier, and that does not allow any toxins or lymphocytes to enter these cells so that they can attack them, or in the seminiferous tubules. If by chance any T lymphocyte reach there, for them, Sertoli cells secrete Fas ligand. Fas ligand is kind of protein. And these ligands has receptor on the T cells. So if they bind with the T cells, this Fas ligand, then T cell automatically by itself get into the process of apoptosis, means their lysosome enzyme release and they start killing their own nuclear material, DNA, and their cells shrink and die. So indirectly, you can say the Sertoli cell produce Fas ligand, which kills T lymphocyte to protect the sperm from the immune response. Okay. Spermatogonia and developing spermatozoa are embedded in nurtures by adjacent cells. They provide nutrition and protect them. Sertoli cell also produce another protein called ABP, androgen binding protein. Why you need it? because in this wall, this spermatogonia needs testosterone. When testosterone bind on these cells receptor, then the second messenger get inside and then activate them for this division. So they need testosterone. So spermatogonia and developing spermatozoa, no, here. So they produce androgen binding protein, and that protein inside the wall of the seminiferous tubule bind with the testosterone and increase the concentration of testosterone in the seminiferous tubules so that spermatogonic cells transform into spermatozoa, or will help the process of spermatogenesis. Are you following me? Sertoli cells also provide negative feedback by production of inhibin. So you saw that, how they do. So what are the function of Sertoli cells? It creates blood-brain barrier. It creates blood-testis barrier, so it does not allow toxins or white blood cell to reach there. They produce fast ligand. They provide nutrition to the spermatogenic cells. They produce inhibin, and they produce androgen binding protein. So if in the short answer question in the exam, if I ask, write two function of the Sertoli cells, you need to know these four. Or which one is not, then you need to know. So the spermatozoa now. After the process of spermiogenesis, we make spermatozoa. Spermatozoa has head, midpiece, and the tail. Head is covered by acrosome, which is an enzyme, and that enzyme helps in borrow the wall of the egg so that it can get inside. So you see here, acrosome is making hole inside the wall of the egg, and then it goes. And to move the sperm from vagina up to the fallopian tube, this is the meat piece. It is like a rotator that rotates and moves it. So tails moves and then it can swim. Once this head get inside, the tail and the meat piece remains outside. This tail and meat piece contains mitochondria. And that's why all the mitochondrial DNA from your father remains outside. It does not get into you. So whatever mitochondrial DNA you get from your mother only. This is the hormonal control of spermatogenesis we talked about. What are the function of the Sertoli cells? And this is written here. We have explained it. Now, male accessory sex organs. Let's go here. Male accessory sex organs. So accessory sex organs are the epididymis. So epididymis is around 6 feet long, 16 feet long, and are non-motile and cannot fertilize. So if the spermatozoa is still in the epididymis, they are non-fertile sperm. They cannot fertilize. And this is because there is low pH. Spermatozoa mature and become motile in the late epididymis, and then prostatic fluid neutralize the low pH and makes them active during ejaculation. So once from the epididymis is passage into vas deferens, then it is motile here in the ampulla of ductus deferens in the ductus efferens. During the male contraception, which is called vasectomy, you see here, you cut and tie the vas deferens so that sperm will not pass up to the ductus deferens. But all other semen and everything will be there, and testosterone hormone will be there. So sexual life and male sex characteristic will be intact, except only one thing, sperm will not be flowing, okay? So that is the vas difference. When it brings here in the ampulla, semen is stored. The vas difference carries sperm into pelvic cavity, and the seminal vesicle here, which is also called seminal gland. This is a kind of exocrine gland. Add fluid which contains 60% of ejaculate. So if there is 100 ml ejaculate, that is 60 ml is the seminal fluid coming from seminal vesicle. This contains a lot of fructose, and the source of energy for the sperm is fructose. Okay. Then vas difference becomes ejaculatory duct, which merge with the urethra in the prostate. Now fluid becomes semen when prostate adds secretion containing citric acid. So it contains citric acid, calcium ions, which coagulate protein. And that's why initially the semen is coagulated, which coagulates semen during the ejaculation. And later the same coagulated semen, when it comes outside in the vagina, then they liquefy again. And that is the function of the prostatic secretion too. So at the beginning they coagulate and then later liquefy it. Okay. So fluid becomes semen when, okay. And then the process of ejection of semen from the penis is ejaculation. But the movement of semen in the duct is called emission. Okay. So ejaculation and ejaculation and emission is the function of erectile penis and the contraction of the tubular wall as well as pelvic floor muscle. And to do that, there must be erected penis. And here is very important mechanism of that. So you have to know the anatomy of the penis first. So if this is the transverse section, you have cut the penis here, and then you are looking from the subject's front. So what do you see? This is the cut stem of the penis. You can see outside there is a skin. Then you have the subcutaneous tissue, and then there is a lot of fascia here. And in that fascia, you see there is a lot of glands, the blood vessel, dorsal vein of the penis. And then you have deep dorsal vein of the penis here. And inside that, there is particularly two group of erectile tissue, which can contract and relax. The superior pair of the contractile tissue is called corpus cavernosa. Singular is corpus cavernosum and two corpus cavernosa of penis. And the lower one is corpus spongiosum. You can see there is a lot of blue structure. That means it is the erectile tissue. The white parts are erectile tissue and blue are the sinus of the veins. There is highly venous structure. In the corpus spongiosum, you see a lot of venous structure and the blood supply. So when during the parasympathetic stimulation, blood flow increase in these veins due to the dilation. And then when you have a tube like balloon and you put water, what happens? Balloon will straight. And that's why when engorgement of these veins increase blood flow in the penis, but return of the vein is constricted. And that's how penis erect. Okay. So erection is controlled by hypothalamus and sacral cord, which is the parasympathetic nervous system, occurs as a result of parasympathetic induced blood flow in the erectile tissue of the penis. What is the neurotransmitter for parasympathetic here? Main is always acetylcholine, but here it is nitric oxide is the neurotransmitter. Erectile tissue include corpus cavernosum and spongiosum. Venous outflow is partially occluded, and that adds to the erection because there is already increased flow and you are obstructing the backflow, like returning. That's why there is erection. Okay. So let's see how, when parasympathetic stimulation starts, how that parasympathetic neurotransmitter acts on the smooth muscle wall of the erectile tissue of the penis and how they erect the penis. So a smooth muscle wall of the blood vessel. Are you following me? Blood vessel histology. So to understand this one slide, you have to understand the blood vessel structure. If you don't understand blood vessel structure, I can say it whole night and day and you will not understand. Okay. So blood vessel has three layers: inner tunica intima, tunica media, and tunica externa. Okay. So sympathetic parasympathetic action release acetylcholine that stimulates the blood vessel inner lining, vascular endothelium, and with that activation they produce, activates endothelial nitric oxide synthesis enzyme, and from L-arginine they make nitric oxide. Or parasympathetic nervous system directly release nitric oxide, whether it is from here or here. They go to the middle wall of the blood vessel in the penis, and their receptors are on the smooth muscle, vascular smooth muscle cell. And what they do, they activate guanylate cyclase that convert guanosine triphosphate into cyclic GMP. Cyclic GMP closes calcium channel. When calcium channel is closes, what happens? That decreases the cytoplasmic calcium. It is calcium which causes muscle contraction. You know that, yes? So if there is no calcium, that means smooth muscle will relax. If muscle relax, blood vessel diameter increase or decrease?
Increase.
Increase. That's why you
Increase.
have engorgement of erectile tissue, and that cause the increased blood flow, and that's why there is penis erection. But we use Viagra. What is Viagra? So do you think this cyclic GMP is forever? If there is no other mechanism in our body, and if every time parasympathetic nervous system come, penis will be for erection forever. That is called priapism, sustained penile erection. And that is very painful condition, which happens in the sickle cell disease. I'm not gonna go there, okay? So to bring it back to the normal condition of these vascular smooth smooth muscle cell, we have another enzyme in our smooth muscle cell that is called PDE. And in this slide, I have written everything here, so you don't need to worry. That phosphodiesterase enzyme change the cyclic GMP into inactive 5-guanosine monophosphate. And this has no effect on these things. So muscle will contract again and its diameter will reduce and penis will be relaxed. In erectile dysfunction, you see, you know, some people have erectile dysfunction, like old people, most commonly in the type 2 diabetes. What happens? Their parasympathetic nervous system is weakened. Their parasympathetic actions are weakened. So in that case, you have less nitric oxide. So less nitric oxide will bring less cyclic GMP. So what do you want to do? You want to keep that less cyclic GMP available for longer time and keep that inside, so it will cause the penile erection. To do that, what do we do? We don't have to kill this cyclic GMP. Who kills the cyclic GMP? PDE, phosphodiesterase enzyme. So Viagra is a medicine called sildenafil citrate. Viagra inhibits this PDE. Once this enzyme is inhibited, whatever amount you have will work for longer time. And that's why the old individual, the type 2 diabetic, dysfunctional erectile dysfunction people can have good life with that medication. But what will be the side effect of this then? this is not on the penile muscle. You have blood vessel all over the body. So if you are taking nitric oxide, the Viagra, that means Viagra will have effect on all the muscle cells a little bit too, other muscles in the blood vessels. So pretty much all other blood vessel will be dilated too. And if you have dilated blood vessel, your blood pressure will drop. Are you following me?
Is that when people get heart attacks or something?
Yes. So what happens? Patient with Viagra sometimes can collapse during the sexual act. Okay. So they can die happily with Viagra sometime. That's why that is the side effect of Viagra. People with erectile dysfunction needs to take the Viagra. If you have cardiovascular problem, then even more with care. Okay. That is here. This is emission and ejaculation. I talked about it, what it is. And then this is male fertility. And this is just the data. So here is the cause: marijuana, cocaine, anabolic steroid can cause male fertility. Or if you are exposed to high heat all the time, that can cause too. So this is all, I think, for today. Thank you, guys.
Thank you, Dr. Shah.
Thank you.
Thank you, Professor. Sorry, Dr. Shah, I just want to clarify that list that you have at the second to bottom bullet point there, marijuana, cocaine, and anabolic steroids, that is that causes infertility, right?
Yes. Yeah.
Okay. Okay. I just wanted to make sure. Thank you.
Yep.
Hey, Professor Shaw.
Uh-huh.
I'm not sure if you have it on the schedule, but am I presenting today?
Judy?
Yeah,
Let me check.
it's just because I was enrolled in the AM lab, so I wasn't sure if you had me down for.
Yeah, AM
Yeah.
lab. Yes, because I think, let me check in my list.
Yeah, it was in the latest email.
Oh,
that
was
you
it?
sent.
You're-
Yeah,
Oh, you see it?
it
Thanks,
shows
Griffin.
everyone's like date and what their topic is and stuff.
Oh, cool. My name's on there.
I believe so.
Oh, cool. Thanks, Griffin.
Yep.
Judy, I have put you on morning lab on 11, May 11.
Morning.
Yeah.
I thought I signed up during the.
Do you want to do?
afternoon
Do you
lab.
want to do here afternoon?
Yeah, I can do it today.
Okay, let's do it
Well,
today then.
Is that fine?
Yeah.
What do you want me to go like last?
Last, yes.
Okay, sounds good. Thanks, Professor Shah.
Thank you.
Thank you.
We'll have 10-minute break and then we'll do it. Okay.
So we're moving over to the weekly, like whatever separate one now, right?
No, I'm gonna give you guys a name. I'm gonna put your name.